therapeutic management of hemoparasitic diseases of sheep and goat and its prevention. He is Dr. Vector Babs. Everybody knows his name because he discovered the Babesia organisms. So that's why today's whole my topic I attributed to him. So generally, sheep and goats rearing are an important source of income for landless poor and the farming community, all of you know. And particularly uncertain vegetarian climate, unpredictable weather, drought, floods, migration of flocks during fodder scarcity, and stress leads to increased prevalence of disease. The small ruminants are highly susceptible to tick infestations and tick-borne hemoparasitic diseases such as anaplasmosis, babesiosis, theileriosis. However, they are often manifested as subclinical infections in case of sheep and goat generally. The distribution of blood protozoan infection in livestock varies with prevailing the climate conditions, vector population, and management practice. And proper understandings of the regional epidemiology of parasitic disease are essential to implement effective intervention measures. Generally, you know that as production of small ruminants usually involves the animals being reared outdoors. They are exposed to a variety of vector-borne diseases that are transmitted by arthropods like ticks, fleas, and mosquitoes. You know that particularly. And among hundreds of bacteria, virus, protozoa, helminths have been found to require hemophagus, particularly blood-sucking arthropods, for the transmission between vertebrate hosts. And as compared to any other group of blood-sucking arthropods, ticks particularly transmit more pathogen species around the world, affecting both human and livestock. So how hemoparasitics, particularly why increasing day by day? You know that recent tick-borne infestations is increasing, particularly in everywhere. In particularly, if I when I go next slides, now I'll show you that how it is increasing day by day. One is global warming. You know that due to that global warming, that the breeding of tick increasing day by day. There are deforestations, and particularly their habitat is lost, and that's why they move from one place to another place. And mutations due to all the factors, particularly due to temperatures, humidity, all the factors may help for their mutations. And insecticides resistance, particularly haphazardly use of insecticides may develop resistance, and as a result, the ticks survival is more. So that the global warming affect the biological vectors, how? Particularly increased feeding frequency, increased egg production, decreased length of development cycles, decrease in extrinsic incubation period, high vector density, increased vector capacity to transmit virus, and high transmission rate there helps for tick infestation. So it indicates that global warming helps for tick infestations. In case of suppose distribution pattern of in case of ticks, in particularly they are particularly in India, that Mondale 2012 he reported that everywhere all over India that tick is scattered, and that's why the tick-borne infestations is increasing day by day. Here also, vector-borne disease mortality distribution also increasing. If you see that all over the world, particularly that vector-borne disease is scattered, and it particularly Africa. You know that Africa is the place where all the diseases are prevalent. In case of Nigeria, also same hemoparasitic disease overall prevalence is there. If you see that particularly 18% in case of sheep infected and 66% is infected in goat particularly. So similarly, in case of Bangladesh also, they also reported the prevalence of that particular hemophagus disease in goat. That is also 25% hemophagus disease affected, particularly that Joel et al. 2020, he reported that particular the prevalence of Haemoprotozoan disease. Similarly, in case of particularly 2000, if you see that India, Pakistan, and that Bangladesh, if you see the 1975 to 2016 to 2001, they are increasing pattern of hemoparasitic diseases. It indicates that due to climate change particularly. So here also same, goat Thaliacea, particularly Kashmir Valley. Then West Bengal, they also reported that particularly Thaliria species, Anaplasma and mixed infection. They also reported that particularly Patra et al. 2018, he reported that Thaliria, Anaplasma and mixed infection was is reported particularly in West Bengal. Similarly, in case of Tamil Nadu also, Thaliria Anaplasma is prevalent, particularly Velusamy et al. 2015, he reported that sheep and goat are affected with Haemoprotozoan parasites, particularly Thaliria and Anaplasmosis. Here also, that particularly prevalence of Anaplasmosis species in India and world, if you compare, you will see that particularly middle our east in India, particularly the 61%, and in Assam here, particularly northeast regions, 16%, though it is written, but one thing that it is increasing now day by day because their research is very less. That's why the reported is also very less. So that's why the particularly prevalence of particular northeast region is showing that very less. But almost all over India are affected with hemoparasitic disease. Almost all India is also affected with Trypanosoma evanse. It is not only that sheep and goat, as a whole, all the animals are suffering from trypanosomiasis. In Assam also, Begum et al. 2018 reported that Thaliria 72.73%, and particularly goat, they are suffering from Thaliria species. It indicates that because it is very rare, in Mizoram also, that Thaliria loanseni infections was also there during 2016 to 2017, that our students' research indicated the presence of that particular Thaliria. So now economic loss, why it is important particularly that it is morbidity, mortality, and case fatality rate, though particularly dog are subclinical from, but sometimes it is very complicated with other diseases due to not proper diagnosis, that animals are mortality is increased, then production and productivity is lost. Once that animal is suffering from hemoparasitic disease, anemia is developed, and due to that their production is going down. Then diagnostic expenditures, it is also increased because particularly through clinical science, it is very difficult for diagnosis only but for that only experienced veterinarian is required. Otherwise, you have to go for blood smear examinations, and during that process, that expenditure also high. Then veterinary treatment, I also that in treatment also it is very, because if you go for treatment, particularly in the Thaliria buparvicin, one dose is costing around 1100 rupees. So for our poor farmers, it is very difficult for there. So that's why veterinary treatment is also again, and control cost of ticks. That is most important, because for prevention of hemoparasitic disease, the control of ticks is the major rule. Then particularly small ruminants, this is some species, Anaplasma ovis, its mortality is very low. Anaplasma phagocytophyllum, I will discuss later. High is very common. Babesia ovis also high and low, both are there. So these are that Trypanosoma also low. But one thing, though wasting conditions, though it is showing that one, but when the tick infestation is high, then fatality rate is also high. And if parasitemia is high, then fatality rate is also high. I will discuss in one by one. So if you see, because for that particularly dynamics, if you go for hemoparasitic disease, particularly susceptible host, vectors, and pathogens, their virulence, according to that, all that particular three factors responsible for particular vector-borne disease dynamics. If you go through, because particularly tick survival, lifespan, reproduction, their biting behavior, all are depend their particularly severity of the infections. Now come to the one by one. I will discuss particularly babesiosis, though in particularly my topics, though I have mentioned that different species of babesiosis, but as a veterinarian, that much is not so important. In field conditions, no need to that good what species is required for diagnosis. If you know diagnosis that it is a babesiosis, then you can go for treatment. That Babesia motasi, Babesia, there is no need to remember all these things, but only for refresh that all the veterinarians, that some species I included here because of that your refresh your memory. And major vectors, that is also not so important. Ticks is sufficient. The ticks is infested, so definitely if tick is infested and temperature is high, you can able to say that the goat or sheep suffering from babesiosis may or may not be. But if history is showing that tick infestation is there, then definitely you can diagnose that it may be babesiosis. So here some same anaplasmosis, particularly Anaplasma ovis, Anaplasma phagocytophilum, Arlesia ruminantum. It caused heart water disease. And Anaplasma capra, that 2018 it is discovered that particularly new varieties there. Say all are particularly transmitted through tick. Then Ehrlichiosis, particularly Ehrlichia bovis and Ehrlichia ovina, they are also responsible for causing Ehrlichiosis in case of sheep and goat. And major vectors, particularly the ticks are the major vectors. Then Theileriosis, particularly Theileria hirsi, Theileria ovis, Theileria loencini. These are some species which are responsible for causing Theileriosis, and major vectors are particularly ticks. Same in case of trypanosomiasis, sheep and goat, Trypanosoma vivax, Trigonosimini, Trigonosimi, Trigonocongolens, Trypanosoma brucei. These are some species, and that are particularly fly. This fly is responsible for transmit the trypanosomiasis. You know, just I mentioned here. So how transmission is occurs, because major vectors is ticks, but it may ticks, particularly in case of babesiosis, Theileriosis, and anaplasmosis, and flies in case of trypanosomiasis. Though it is the major vectors, but sometimes during blood transmissions also it may transmit from one animal to another animal. Through needles also it may transmit. So these all that particularly flies, mosquitoes, then the needle during castrations, blood transmissions, all are responsible for transmission of hemoparasitic disease from infected animals to healthy animals. So now come from babesiosis. From one by one I will discuss here. So you know that the disease babesiosis results in a heavy economic loss to the small ruminant industry, particularly in the tropical and subtropical countries. In India, the estimated loss due to tick-borne disease is around 498.7 million per annum in dollars, and in the world it is roughly $7,000 million. You see, it is not only that sheep and goat as a whole animals are responsible for that. Particularly, Babesia ovis and Babesia motasi are pathogenic, and Babesia ovis is smaller in size. And the classical presentation is febrile syndrome, how you know, along with anemia and hemoglobin urea. That is the most important clinical signs for your diagnosis. I'll come when I'll discuss in the diagnostics part. Ovine and caprine babesiosis occurs in all breeds and sex of sheep and goats, but animals 6 to 12 months old have a higher incidence than animals of other age groups, because small animals are, because young animals are mostly developed babesiosis due to low immune. And in case of babesia, particularly pathogenesis, how disease occurs, if you know that one, just I will refresh your memory that babesia produces acute disease by two principal mechanisms. One is hemolysis and circulatory disturbance. During the tick bite, sporozoites are injected into the host and directly infected red blood cells. And during that particularly they infect erythrocytes, and resulting in 2 or sometimes 4 daughter cells develop, and they leave the host cell to infect other erythrocytes. And during that process, they cause intravascular and extravascular hemolysis. When lysis dividing parasites in the red blood cells, they produce rapid destruction of erythrocytes with accompanying hemoglobinemia, hemoglobinuria, and fever. During that process, fever will support. Then overproduction contributes to disease progress, causing vasodilation, hypotension, increased capillary permeability, edema, vascular collapse, coagulation disorder, endothelial damage, and circulatory stasis. that I will show in the pictures. If you see the pictures, particularly the tick bite, when particularly that babesia organism develop into the tick, and when they bite to the animals, they directly convert to the circulatory, and in the blood cells, they from blood they multiply it and rupture the, if you see the picture, rupture the erythrocytes. It may lead to development of intravascular hemolysis, and during that intravascular hemolysis, then hemoglobin is released. As a result, anemia is developed. When anemia is developed because of due to low oxygen carrying capacity, so as a result, all the body functions, particularly whatever the organs, their body function is goes down, and they may cause, in case of jaundice, because you know that release of hemoglobinuria, and it may lead to development of jaundice, and hyperbilirubinuria, bilirubin level is increased, and hepatomegaly that may cause, and in case of kidney, it may cause hemoglobinuria or hemosiderin-uria, particularly in case of, and particularly that babesiosis, if you know that it is called hemoglobinuria because of intravascular hemolysis. So clinical signs, that is the most important factors for diagnosis. Not only that babesiosis, all the body function is goes down. That's why that everything may be possible, particularly fever. You know that because during when babesia is entered into the body, that phagocytic actions or defense mechanisms may active, and during that process, temperature is increased. Animal may become lethargy because of hemoglobin level goes down. Varying degree hemolytic anemia, I already mentioned. Anorexia, presence of ticks also, that is also one of the factors. Pigmenturia, weakness of collapse, pale mucous membrane, because you know that the hemoglobin level goes down. Icterus, splenomegaly, lymphadenopathy, then tremors, because tremors in particular cerebral babesiosis during that. Acute kidney injury, not I am really acute respiratory distress, because all the body function is goes down. That's why you can myocardial dysfunction also, all the body function goes down. And neurological signs, particularly babesiosis, is all the nervine symptoms may also develop, and that's why that disease may complicated with polyencephalomalacia, particularly dog, sheep and goat are mostly suffering from polyencephalomalacia due to thiamine deficiency. So, but in that case, if temperature is there and nervine sign is also there, and tick infestation is also history is there, you can assume that it may be not due to polyencephalomalacia, it may be due to babesiosis. So particularly, I already mentioned Babesia ovis and Babesia mortacea is the most pathogenic Babesia organisms. So, and Babesia carcass, that presence of anemia is very less, sometimes may develop also, but pathogenicity is less. So here some pictures I read, particularly I have seen from that one, the icteric sclera, and oral mucosa membrane of Babesia infected goats. So here also it is not, again, I am telling for differential diagnosis. It is not that only that when pale mucous membrane, if you see, it is not only due to babesiosis. For babesiosis, rise of temperatures, then pale mucous membrane and history of tick is essential. Otherwise, suppose due to maybe parasitic infestation also, Haemoncus, that most important that no, so they may also responsible for causing pale mucous membrane. So hemoglobinuria, that is most important factors. When condition is so severe, last part of the disease, they develop hemoglobinuria. And that is also other for differential diagnosis, leptospirosis, that is also one of the factors. Postprandial hemoglobinuria, bacillary hemoglobinuria, they may also responsible for causing hemoglobinuria. But for that, for bacillary hemoglobinuria and babesia, it is very difficult for diagnosed by the clinical symptoms. You have to collect the blood samples and go for smear, and then you can able to diagnose whether it is due to bacillary hemoglobinuria or due to babesiosis. Laboratory findings, hemoglobin level goes down. I here one case I attended, and that's the hemoglobin level is 4 gram percentage I've seen, and PCV is 10%. So particularly postmortem findings, it is a dark kidneys and jaundice that carcass in case of babies in sheep, and enlarged spleen in case of babies in sheep. Particularly if it's particularly enlarged spleen in case of other disease, particularly anaplasmosis, theileriosis, also you will get in enlarged spleen. And particularly in all hemoparasitic diseases, when you open the carcass, it is showing anemic. Now come to the anaplasmosis. It is also known as goat sickness, the infectious hematopoietic disease caused by several species. I already mentioned that Anaplasma marginally, Anaplasma ovis, and Anaplasma phagocytophilum. There are huge economic loss also, and the prevalence of anaplasmosis was found significantly higher in Jamunapari and crossbreed in compared to Bengal goat, particularly Rahman et al. 2022. He reported that one, that Jamunapari and crossbreed. Bengal goat is an indigenous breed. That's why sometimes the indigenous breeds are more resistant to hemoparasitic disease than the crossbreed and pure breed sheep and goat. They also reported that the study revealed that prevalence of anaplasmosis was higher in goats not using acaricides. You please remember that those farmers are using acaricides, the chance of hemoparasitic disease less than those who are not using acaricides. That if you is always, when those who are using acaricides, the hemoparasitic disease chance is very less. Regarding tick infestation, the prevalence of anaplasmosis found significantly higher in tick infested goats. That is also because sometimes what happens, non-tick infested goats, but whatever disease is transmitted, I already mentioned it may be due to tick and maybe due to other factors also made responsible for causing hemoparasitic disease. But those who are infested with tick, their chance is vary. It does, however, have more often goats than sheep. Sheep are less commonly affected anaplasmosis than goats. So I already mentioned that one, how is transmitted in my previous slides. So pathogenesis, I already mentioned here also, same thing in case of anaplasmosis ehrlichiosis, particularly when they bite, particularly then it, particularly monocytes are mostly affected with Ehrlichia that enter leukocytes by phagocytosis, and due after the formation of phagocytic vacuoles, and that bacterium survive and multiples in phagosomes, and after that cell and phagosomes lyse, and they cause particularly circulating in the and vascular intravascular coagulation is occurs, and as a result they may lead to development of anemia and thrombocytopenia. You please remember, if suppose a differential diagnosis babesiosis and anaplasmosis, anemia in both the cases, but thrombocytopenia mostly occurs in case of your anaplasmosis and ehrlichiosis. And due to that anemic conditions, same liver is enlarged and it may cause hepatopathies. So almost in all the your hemoparasitic diseases, their mechanism of actions or pathogenesis are almost similar. Then clinical findings, particularly here, or everything, if you go through that, everything is similar. Progressive hemolytic anemia, pyrexia, loss of weight, weakness, anorexia, constipation, dehydration, jaundice, labored breathing, depression, abortion, all are same with babesiosis also. But here only you can able to say due to thrombocytopenia. Anaplasma phagocytosis infect the neutrophil granules and cause tick-borne fever in sheep, with the most common symptoms comprising high fever, loss of appetite, dullness, and decreased milk yield. Here it is a paper white mucus membrane. In case of babesiosis also you get tick infestations, and particularly nervine symptoms I already mentioned in case of babesiosis also. In case of here also due to particularly anemic conditions, good depression, tick bite, only pale oral mucosa, yellow coating of the tongue, and monocytes. I will in the next slide I will show how to diagnose that particularly. And particularly heartworm infections, that is your ehrlichiosis, got the neck is extended, consistent with dyspnea. It may be also that PPR, one of the factors also showing that same symptoms, particularly your respiratory distress and dyspnea. And Postmortem findings, I already mentioned in the baby case of babesiosis. Here also, you'll use discolorations and consequent enlargement of the spleen. In case of babesiosis also, on left compared to the normal spleen, if you see that it is most, particularly here, it is enlarged. This is called splenomegaly. And clinical pathology, same with hemoglobinuria, platelet count. Here, this is the packed cell volume, thrombocytopenia. You please remember that thrombocytopenia is the most clinical cardinal signs or clinical pathological signs in case of anaplasmosis and ehrlichiosis. And AST, ALT, and AP is increased, hypoproteinemia, hypoalbuminemia, and hyperglobulinemia. Then comes the theileriosis. Theileriosis, small ruminants, is caused by same. I already mentioned that Theileria lastoquadri, particularly it is a malignant ovine theileriosis. Theileria ovis is a benign theileriosis. Theileria recondita is causing mild ovine theileriosis, and some other species also they are also responsible for causing theileriosis. It is a same major threat to livestock industry, and that this is also important due to its significant international trade of animal and animal producers, and particularly in India, that Theileria hirsi and Theileria ovis are the most prevalent species reported in small ruminants. Particularly pathogenesis, also that also same in case of babesiosis. Within host leukocytes, the parasites induces leukocyte cellular divisions, which expands the parasitized cell populations, and infected cell disseminated throughout the lymphoid systems via the lymphatic and blood vessels. The infected leukocytes may block capillaries, causing tissue ischemia. That is, here I mentioned here lymphoid systems. That's why babesiosis, then anaplasmosis. In that case also, particularly lymph node is enlarged, but theileriosis is the cardinal signs that prescapular lymph node enlarged. You please remember, with that, if temperature is high, prescapular lymph node is enlarged, and thick infestation is there, you can diagnose that it may be due to your theileriosis. Clinical, here I already mentioned a rise of temperature, enlarged lymph node, that is the most important. Then inability to walk due to pain due to enlarged lymph node, and when you palpate, the feeling pain. Then anorexia, listlessness, moderate anemia, nasal discharge, edema of eye, and corneal opacity. Though it is corneal opacity in case of babesiosis also, because I already mentioned when anemia is developed, all body functions goes down. That's why that all the symptoms you can see in any hemoparasitic diseases. I already mentioned here pathogenicity level, that if you go through that particularly your Theileria ovis, in case of that immunocompromised and animals, this is maybe subclinical form, and particularly that hemoparasitic disease mostly in subclinical form in sheep and goat. But when tick infestation is more and their pathogenicity more, it may goes to the fatal conditions. Here also, theileriosis, pale pink mucus in growth. I already mentioned in anaplasmosis also, babesiosis also, and intramandibular edema in caprine theileriosis. You please remember, intramandibular edema in caprine theileriosis is a cardinal sign, along with your prescapular lymph node in case of goat, enlarged prescapular lymph node. That is also one of the most important clinical signs for diagnosis of your theileriosis. The same, your particularly postmortem, photograph of heart showing yellow fat indicating jaundice, photograph of prescapular lymph node, that is enlarged prescapular lymph node, then gallbladder is particularly enlarged gallbladders, and particularly photograph of spleen showing splenomegaly. That I already mentioned that babesiosis, anaplasmosis, ehrlichiosis, and theileriosis, in all the conditions, the spleen is enlarged. Then come to the trypanosomiasis. It is also one of the most common hemoparasitic diseases that is called Haemoprotozoan. You can show Haemoprotozoan. The total economic loss due to SARA among goat was estimated to be Indian currency 340.88 million, including sheep and goat, then all other animals also. Major losses due to reproductive problems, reduced milk, and meat yield particularly. And natural infection with Trypanosoma congruens, Trypanosoma vivax, Trypanosoma I already mentioned, and Trypanosoma evensi have been described in case of goats. And trypanosomia goats produce acute, subacute, chronic, or subclinical forms, particularly in case of goat and sheep. In Asia, the name of SARA is mostly employed, although several other names were used before, such as Purana, the chronic or old, Tibersa, 3 years disease, and Dubla, emaciated, or Makiki Bimari, that is known as in case of horsefly disease, but you can say in case of sheep and goats, Makiki Bimari means is transmitted through mosquito. Goats may also serve as a reservoir of trypanosoma infection for other species. You please remember, including the human beings in case of Trypanosoma brucei rhodesians. That's why it's the most important for diagnosis of trypanosoma in case of goats. Goats can play an important role in the dissemination of the disease. Thus, goat necessarily infected with Trypanosoma congruens, Trypanosoma vivax, or brucei, and presenting clinical disease are regularly observed in Africa. But in India also, we can regularly monitor that infection of Trypanosoma because the goat act as a reservoir. So it may transmit to other species, including human also. So I already mentioned that one is not so important. Epidemiology, worldwide, I already shown that is in India, SARA occurs mostly in post-rainy seasons, August to October. So not only Trypanosoma, for it is same in case of other factors also, because during that is the most suitable climate for breeding of all the fly and mosquito and ticks also. And when the breeding is and lowest incidence in April and May, because during that the temperature is high. Pathogenesis, I already mentioned the main pathogenic mechanism of anemia include hemolysis, red blood cells, sequestration of hemodilation, platelet aggregation, lipid peroxidation, bleeding, and bone marrow dysfunctions. I will show here, if you see that particularly trypanosoma, in case of blood, then through lymphatics, and it go through central nervous systems. So here, if you see that from temperature, high rise of temperature, increased lymphadenopathy, anorexia, that definitely, and other nervine symptoms. So that's why I am telling that hemoparasitic disease is very difficult for diagnosis, whether it is babesiosis, whether it is anaplasmosis, whether it is ehrlichiosis, whether it is theileriosis, whether it is trypanosomiasis. But it is, if suppose any indicate that rise of temperature, tick infestation is there, in that case, you should go for blood smear examination. Then you can able to diagnose whether it is babesiosis, ehrlichiosis, anaplasmosis, and trypanosomiasis. So here the clinical observation, particularly clinical symptoms, particularly rough hair coat and uncoordinated postures, that is their nervine symptoms, and severe scrotal edema. This is one of the major clinical signs, scrotal edema. Here also scrotal edema and your poor coordinations. Then corneal opacity, that is also more, it may be unilateral, it may be bilateral. Though it is sometimes that corneal opacity may be other problem also. But if suppose rise of temperature and tick infestation and corneal opacity is there, you can 99% able to say that it is a trypanosomiasis. Same that I'm not going for postmortem because all are same in case of babesiosis, anaplasmosis, all in a hemoparasitic disease, that particularly that the postmortem findings almost are similar. Now come to the most important part is diagnosis. How you diagnosis. I already mentioned that particularly mostly through clinical symptoms, but experience is required. With that, you can able to say that animal is suffering from babesiosis, anaplasmosis, theileriosis, ehrlichiosis, and trypanosomiasis. But all the disease, particularly in case of babesiosis, the most cardinal signs is hemoglobin urea. In case of anaplasmosis and ehrlichiosis, I already mentioned that thrombocytopenia, which may lead to development of epistaxis, bleeding from the nose, though it is very rare in case of sheep and goat, but sometimes it may happen, and in that case you can able to say that goat is suffering from anaplasmosis and ehrlichiosis. In case of thylarosis, I already mentioned that most important clinical signs is prescapular lymph node enlargement. And in case of trypanosomiasis, if corneal opacity, high rise of fevers, and thick infestation is there, history is there, then you can able to say it is your trypanosomiasis. Then you have to go for particularly collect the blood samples. Here you can collect the blood sample from jugular vein also, or particularly from your tip of the ear, particularly when from tip of the ear, one drop of blood, you take it in the microslides and prepare smear, then go for Giemsa stain and see under microscope. That is the most important for diagnosis of, particularly in case of field, because in field only that is you can able to do it. So particularly babesiosis, there are different forms there. One is pyroform, round, leaf-shaped, and ovoid to round in shaped. By the though it is I am written Babesia motacis, Babesia ovis, but as a veterinarian, you know only that it is Babesia species. If you do that, then it is confirmed. I will show up some pictures. Microscopy demonstration typical intracytoplasmic in case of Echidnas morula, initial bodies in case of your or anaplasma phagocytophyllum. Yeah, I will show you that pictures also. Then here morula is there, and particularly in case of thylaria, pyroplasm or cox blue body. I will show you that some thylaria and trypanosoma. You know that is very easy for diagnosis because it is a extra that muscular space it may remain. So here you see that pictures. Here, if you see that particularly, some are oval shaped, some are round shaped, and some are dot shaped also. That is indicate that presence of Babesia organisms. There are different types of this there, so mostly that is there. Then another one, though I am mentioned Giemsa stain. Another one stain is there, acaridine orange stain. That is very easy, and fluorescence microscopy. If you see that, particularly if you see pictures indicate that presence of Babesia organisms. It's very easy for diagnosis. So here, particularly Giemsa inclusion body, okay, anaplasma bovis and ovine monocytes are visible. If you see, all are monocytes. Actually, Ehrlichia and anaplasma, previously known as a ehrlichiosis, but due to the inclusion body presence, different blood cells, that's why their name is monocytes. If it is there, it is known as ehrlichiosis, and if present in the neutrophil, the inclusion body present in the neutrophil, then it is known as your anaplasmosis. All are ultimately all are same. Is the inclusion body. Sometimes when that inclusion body clumps together and from morula, in case of particularly here, it is your neutrophil and presence of your anaplasma organisms. If it is monocytes, if it is, then it is known as ehrlichiosis. Here also inclusion bodies are there, so it is a neutrophil contents. Then come to the thylaria, you know the cox blue, the lymphocytes, presence of inclusion body. It is known as cox blue body, or you can in the RBC also, it may be there particularly in the, there's also is a cox blue body. And particularly trypanosoma is very easy for diagnosis. You can just you take one drop of blood and see under microscope, and their motile your trypanosoma is visible. And otherwise, if you go for Giemsa stain also, it's very easy. That one. So Napoleon had that everything is there. So it is very easy for diagnosis. Now laboratory findings, I already mentioned that one. In the case of babesiosis, all parameters are goes down, particularly hemoglobin, RBC, then packed cell volumes, then lymphocytes, monocytes are significantly increased also. and particularly total protein level is elevations, then significant elevation of burn, creatine, cholesterol, triglyceride, HDL and LDL level also. Anaplasmosis also you are getting, but most important is thrombocytopenia. Then failurosis is also, all are same in case of babesiosis is also. Then trypanosomiasis, leukopenia is the most important characterized, followed by neutropenia, eosinopenia, and lymphocytosis. But one thing you please remember, only for when you go for laboratory diagnosis, means evaluation of blood samples and serum samples for hematobiochemical analysis, you can correlate it with your symptoms, history, and blood smear. Then you can able to diagnosis. And other factors, the animal inoculation test, serological test, IFT test, ELISA test, and most that the diagnostic value is molecular test. Now come to the therapeutic measurements. Though you know everything, what treatments you should provide particularly. In case of babesiosis, generally we are diaminogen acetate and imidocarb dopamine are generally used against babesiosis. But sometimes what happens nowadays, that diaminogen acetate and is having some toxic effect. That's why researchers are showing for new drugs, particularly in that case, they are using combined, particularly combined therapeutic measurements. In case of particularly, you can use azithromycins or bupropion, along with that enrofloxacin also. Is nowadays 3 or 4 combination drugs are using for control the babesiosis. Though we are using diaminogen acetate, what happened that they never eliminate the organisms for whole life. It is not possible. That's why people are using. But though I am here mentioned clindamycins, generally not to use in case of sheep and goat because it may sometimes develop the diarrhea. But most important drugs is your the imidocarb. Instead of diaminogen acetate, you better to use imidocarb for control of your babesiosis. Here I want the diaminogen administered at a dose rate of 3.5 mg per kg body showed 80% efficacy at day post medication, and imizoles administered group B animal at a dose rate of 2 mg per kg body weight showed 60% efficacy at day 3, 90% at day 7, and 100% at day post medication. So if you see there, if you compare with diaminogens and imidocarb, it shows imidocarb shows better results than your diaminogens. The combination of imidocarb, dipropionate, and oxytetracycline was highly effective for ovine babesiosis. Please remember, because sometimes what happens, along with, because nowadays I already mentioned, due to eliminations of the organisms from the body, singly only imidocarb or diaminogen may not be possible. So you have to use combination with oxytetracycline. Here one I am done caprine babesiosis in case of your therapeutic hemoglobinuria. I'll provide imidocarb, given imidocarb, and after that for 72 hours that urine is showing normal, if you see here, due to use of your imidocarb. Then side effect, I already mentioned, most of the babecidal drugs available today in the market have problems due to toxicity and development of resistance parasites with that. Diaminogen is not approved by Food and Drug Administration in USA. It is already been, now in USA it is banned. In many countries like Japan, the production of diaminogen itself is stopped. Only in India it is going on. That's why I advised not to use diaminogen acetate. Instead of diaminogen, better to use imidocarb dipropionate along with your oxytetracycline. In case of ehrlichiosis and anaplasmosis, the drug of choice is doxycycline, best method of treatment, 10 mg per kg, and strong long-acting oxytetracyclines also you can able to use. Imidocarb also successful. Then chloramphenicol, that is also useful. Sulfapyridine also you can use. sulfamethoxam also able to use, androfloxacin also able to use, rifampicin also you can use. Actually, what happened that ehrlichiosis and anaplasmosis is rickettsial. That's why you can able to use that particular drugs. Then in case of trypanosomiasis, particularly the recommended drug of trypanosomiasis, buffalo, cattle, sheep, and goats is diaminogen acetate with dose rate of 7 milligram per kg deep intramuscular. You know, other drugs, isomethamidium chloride or melasromine hydrochloride may be used at a dose rate of 0.5 mg deep intramuscularly, and other octos. But one thing, when you give that particularly diaminogen acetate in case of, because in imidocarb also you can use it for that one, folic acid and cyanocobalamin can hasten the recovery in trypanosomia infected in animals. So along with that, you have to use folic acid and cyanocobalamin injections. So these are some drugs you already knows that particularly you used with different routes, and sometimes it may be used by intramuscular or subcut, or sometimes it may be used intravenous. In case of theleuresis, you know, the most common drugs or drug of choice is provaquin and bupropion, and imidocarb have been used for theleuresis treatment. But I already mentioned that bupropion, though it is a drug of choice for that, but cost is very high. So instead of that, you can able to use oxytetracycline as a higher dose. You have 20 mg per kg body weight intravenously with normal saline with vitamin B complex may give successful results. The goat infected with Theileria hirsi was successfully treated with metalcapine 500 mg sulfamethoxyrazine and 25 mg pyrimethamine given in one oral dose with supportive treatment B complex, paracetamol, and feradol. In particular, Banerjee et al. 1990, he used that one and he got successful results. And in my my experience, I used that oxytetracyclines with higher dose. It shows better results along with vitamin B complex. Some other therapy also there, particularly nowadays research is going on botanical herbal treatments. Flower extract of Calotropis procera was effective for the treatment of Theileria in goats, 0.3 mg per kg body weight, orally up to 5 days daily. That Babar 2000, he study revealed that use of herbal drugs. So other supportive therapy, particularly along with that drug of choice, you have to give some dehydration is there, some appropriate field therapy. If animal life-threatening severe anemia, blood transmission, then nutritions also you can able to use to increase the platelet numbers in case of anaplasmosis and ehrlichiosis. Iron supplements is also important, and short course of dexamethasone reported to be successful in controlling initial epistaxis in case of anaplasmosis and ehrlichiosis. Treatment of vitamin B complex you have to give. Then therapy with levamisole, particularly it acts as a immunomodulator, and in case of it, in pancytopenia means when thrombocytopenia count is going down, in that case also you can able to use levamisole. Diuretics, particularly in hard water, in case of ruminants, you have to use for as a supportive therapy, particularly in case of hard water. Now come to the control, how you control, because I already mentioned vectors are responsible for transmission and causing of hemoparasitic disease. So that's why you have to control the tick, particularly manual removal. You have to remove by manually. The burning the patchouli surrounding area, you have to whatever is there. Then biological control, ants, because sometimes that ants may take the ticks. Then predatory mites also might also there. They may also responsible for engulf the ticks. Then chickens. Then you know that I have to ox pickers. They are also helps when the animals are grazing during that time. They take the ticks. Then entomopathogenic nematodes. If you see, they are also responsible for killing the ticks. Then biopesticide, that is also one of the factors for important factors. Nowadays, people are using that biopesticides for control the tick. Then acaricides, you have to use the acaricides, lots of acaricides in the available in the markets. So you have to advise the farmers to 3 times in a year we have to use the acaricides for control the tick. Then particularly Safi and Zayed 2002 examined the acaricide effect of plant extract of neem seed oil. Nowadays that is more because there is no any side effects. So people are using neem seed oil to control the tick. So in particular, though in India it is not too much popular, the tick guard plus the vaccines for tick controls. Other vaccines are available, but some are now only research purpose is going on. Though it is not available in India also, only research purpose is going on. So last conclusions, the effect of hemoparasitic disease causes a huge economic loss on sheep and goat industry worldwide. Hemoparasitic disease rank high in terms of their impact on the livelihood of resource poor farming communities like India. Decreased outputs of animals products, byproducts, manures, etc. contribute to production and productivity losses. Proper diagnosis will help to take appropriate measures. That is most important, because hemoparasitic disease is very difficult for diagnosis, only clinical signs, because that clinical signs is similar with other diseases also. That's why you have to go for proper diagnosis with the blood smear examination or other diagnostic methods. Proper use of acaricides, that is also important to control the vectors and prevent the hemoparasitic infestation. And though the lack of effective vaccines against the tick-borne disease or hemoparasitic disease on man and animals force us to look into strategic control of tick vectors in an integrated format for the effective control of tick-borne disease. Though tick-borne disease are most important because nowadays due to global warming, but that governments should take rules for research purpose to give some, because nowadays I have seen that prevalence and epidemiological study is not so important in instead of molecular diagnosis, but we should go for epidemiological study because we know the pattern of disease prevalence in particular regions. Then it will be easy for diagnosis and take control measures. Thank you very much.